Oh my God, I'm so sorry. I had no idea. I had no idea. I told you not to trust me with technology. You have a question? What's up? No, doesn't count towards your grade. But I like to see it because I like to know who's here. Well, then fill it in. Then fill it in. All right, hopefully it's recording. Of course, because I was trying the iPad, I forgot my little clicker. It's okay. We're gonna make it work. All right, so now we are done with muscle. And we're beginning our last unit, which is on the nervous system. So we saved some of the hardest stuff for last.
Based on the practical,
I think so. Yeah, the practical isn't as, isn't as difficult. So, whatever. So here's a little funny cartoon for you. Hopefully this isn't the case for you, especially if I see you next semester. You know where to hide. So this last unit has 4 chapters. This first chapter is a toughie. So I'm glad you're here today. Be sure you can come next Tuesday if you can. If not, make sure you watch that video. This nervous tissue chapter is talking about nerve impulses, how they're generated, what different types of nerve impulses there are, all that good stuff. And then we'll go into the central nervous system. And these last 2 chapters are shorter. Okay? All right. So here we go. We're going to jump right in. Before we get into the nerve impulse, just a couple quick reminders. Make sure you review the functions of the nervous system. You know this. I know you do. I'm just framing it in this way of a negative homeostatic feedback loop. Bam! Bringing it back to chapter 1. That's pedagogy right there. So basically, we know that the nervous system is the processing, right? This is the brain is overseeing. The brain is overseeing everything. Well, it only can do so because it's constantly receiving sensory input. So we have to make sure we're looking at all of this. The brain is always receiving sensory input. Sensory input from the skin, from the joints, sensory input from the organs. Easy example, if the receptors in your stomach wall, you haven't eaten for a while, your stomach will get a little smaller. So the sensors, the receptors in the stomach wall will sense, hey, there's less stretch in the stomach. And the sensors in the stomach wall will communicate to the brain. The brain will process. Hey, we need food. And then the brain will initiate a motor output response. In that case, it'll send messages to your hypothalamus to make you feel hungry. It'll send messages to your mouth as you think about food. Saliva will start to be all these things. So it's always going to be in this pattern. But that makes sense, right? The brain wouldn't be able to process and send out an appropriate response without getting feedback from the body. I don't know why the Merriam textbook chooses the most lame example. Is this not the most lame example? There's a dude looking at a glass of water. And he's thinking, that water looks good. So the sensory input is visually, he's seeing the glass of water. That looks good. Maybe there's other sensory input from his stomach because it's less stretched. From the hypothalamus, his thirst center. That sensory input goes to the brain where it's processed. And in this case, the motor output goes to the skeletal muscles that will pick up that delicious glass of water. Okay. When we talk about the organization of the nervous system, we have the central nervous system and the peripheral nervous system. This is probably a review for some of you. The central nervous system is the brain and spinal cord. That's it. Central nervous system, brain and spinal cord. The peripheral nervous system is every other nervous tissue outside of the brain and spinal cord. Particularly for the peripheral nervous system, we think about all the peripheral nerves. When we think about this separation, we can also add the functions. If the central nervous system is the brain and spinal cord, what is the function of the central nervous system? Processing, oversight, control. The central nervous system, the brain and spinal cord, that's where we do the processing. And by the way, we can do some processing in the spinal cord. We can process some reflex information. Versus the peripheral nervous system, it is made up of axons. And what do the axons do of the peripheral nervous system? Simply transmit nerve impulses. So the function of the peripheral nervous system, because it's made up of the axon, the peripheral nervous system just transmits nerve impulses. There's no processing happening in your radial nerve. These peripheral nerves are really just there to send nerve impulses. I think that makes sense. Yeah? Okay. You can also look at this in a schematic. And I actually really like this one. So we can see the central nervous system and the peripheral nervous system. Boom. We can further subdivide the peripheral nervous system. So for this part of the lecture, we're only going to go up to here. The rest we'll cover later. But make sure that you are familiar with the two basic parts of the peripheral nervous system. If we look at those peripheral nerves, either it's a nerve sending information to the spinal cord and brain. That would be a sensory or afferent division. Sensory or afferent is when we are conducting nerve impulses from the periphery to the spinal cord and brain. And because it's conducting impulses towards the central nervous system, these are the sensory nerves. Yeah. And then the other half would be the motor division or efferent. This is where we're sending nerve impulses in the other direction from the brain and spinal cord out. Here's where spelling matters, right? Afferent versus efferent. One letter difference. I think about E for exit. So the efferent nerves are exiting the brain, going out. Good. We're going to talk about the rest later. Alrighty. This is saying the same thing, right? Sensory nerves transmit information from the periphery to spinal cord and brain. Motor or efferent send nerve impulses from the brain out. Yeah. Because each individual axon is a one-way street. So each individual axon can only send information in one direction. Our nerves, our peripheral nerves are bundles, so they're going to have a lot of axons, but each individual axon is a one-way street. It can only transmit information in one direction. It's exciting, isn't it? Okay. So I'm going to move pretty quickly through the anatomy because we'll talk about the structure of a neuron. That's not so bad, folks, is it? Cell body, dendrite, axon. Because I want to spend most of our time today on the nerve impulse stuff. I always forget to put this picture in here. So let me go pull it up real quick. I'm going to go back to the tissues. Let's remind ourselves of what the nervous tissue looks like. You remember? Does this feel like forever ago? Okay. So when it comes to nervous tissue, we have two things that make up nervous tissue, right? We have the neurons. These are the nerve cells. So the star of the show are the neurons. They have that kite-shaped body with the one axon. Yes, yes, yes. But we also have a whole host of helper cells, otherwise known as neuroglia. Some say neuroglia. I say neuroglia. Everyone forgets about the neuroglia, but that actually makes up pretty much half of our nervous system volume. So it's pretty, you know, we have a lot of it. So in our picture of the nervous tissue, we see the neurons, obviously, right? But you see all those dots in the background? Yes. Those are the cell bodies of the neuroglia. So we have to consider both because both play a role. The neurons are what transmit nerve impulses, but the helper cells help. I don't know how you all wear glasses with these things. So let's talk about them. The excitement. All right, the nerve cell. Let's do a quick anatomy review. The nerve cell, here it is. We know that it sends nerve impulses. Great. We know that nerve cells live a long time. It's not like the cells of our skin, remember? The cells of our skin that work their way up the epidermis and then fall off. No. Our nerve cells pretty much live as long as we do. I want to point out this bottom bullet point. Nerve cells have a very high metabolic rate. They are thirsty. So muscle cells have a very high need for oxygen. They have a high metabolic rate, and so does nerve cells. So do nerve cells. Good. Anatomy 101, are you ready? Even if you weren't, you're not going to say anything probably. That part of the semester, isn't it? I know. So the cell body, that's the cell body. It's also known as the soma. It's going to have the nucleus. Okay, great. We're going to keep it pretty simple here. Cell body, otherwise known as soma. Easy. Coming off the cell body are these finger-like projections called dendrites. These are the feelers. The dendrites are constantly taking in sensory information. Perhaps sensory information from other neurons. The feelers. Okay. Then we have the axon. The axon is the one long tail. Check it out. We already know that, probably. There's usually only one axon per nerve cell. This is where the nerve impulse is conducted. This is where the electric current. There are other structures and regions, but you don't have to know the axon hillock. You don't need to know the axon terminals. They're there if you want. The axon hillock is just the thickened area in the cell body right before the axon. We don't need to know that. The axon terminals, those are the tendrils. You don't have to know it, but I'm just throwing it back. This is high level teaching, folks. Do not try it at home. Because I referenced chapter 1, I referenced chapter 4 with the tissues, and now I referenced the skin, chapter 5. And now I'm referencing chapter 9. Remember those tendrils? We talked about a motor unit. When a nerve comes down, the end of the nerve has those tendrils that wrap around the muscle. Those tendrils are terminal branches that lead into the axon terminals. But you don't need to know those. And then we know, of course, already from chapter 9, we know that when a nerve connects with something else in the body, it doesn't connect directly. There's a gap junction. Another chapter 4 reference. It's a synapse, it's a space. What kind of chemical crosses the synapse? A neurotransmitter. So a neurotransmitter is the chemical that crosses the synapse. We happen to mention acetylcholine, which is the neurotransmitter that always goes into skeletal muscle. But we're going to begin to introduce many other neurotransmitters in chapter 12. You don't need to know, I mean, because I don't think it's worth anything, really. The presynaptic cell is before the synapse. The postsynaptic membrane or postsynaptic cell is after the synapse. Don't have to know it. What I do want to point out, though, I want to point out that we have neurons connect with a bunch of things. So we talked about, thus far, we probably have in our mind that a nerve goes into a muscle. But check this out. Sometimes a nerve will contact another cell body. And that's a axosomatic synapse, because it's a synapse between the axon and the soma. You don't have to know these, but I'm just making a point that these neurons contact a lot of things. They may contact another neuron cell body, another neuron's dendrites. They may go into a muscle, an organ, liver, kidney, heart. They get around. You're going to wake up when we get to the nerve impulse. Yeah.
The axodendritic synapse, no, we have to move up.
Axodendritic would be this one up here. It's the synapse between the axon and the dendrite. Yeah. Let's talk about those neuroglia. These are the helper cells. There's helper cells in the central nervous system, and there's helper cells in the peripheral nervous system. I am asking you to know one example from each, and they're the starred ones. So even though I'm going to give you the definition of what each does, the only ones you have to know are the astrocytes in the central nervous system, the helper cell in the central nervous system, astrocytes, and the helper cell in the peripheral nervous system called the Schwann cells. It's not the Schwann truck bringing your ice cream and steak. Do they still have Schwann trucks? They what? I just saw one today. Oh, see? All right, so you can see here, this is kind of a cartoon-like picture. You know, we saw the nervous tissue where you just saw the dots. You couldn't really tell much about the neuroglia. Here are some examples of the central nervous system neuroglia. You don't need to know these. It's interesting though. These helper cells do a lot. You can see the ependymal cells. They produce cerebrospinal fluid. Oligodendrocytes, they form the myelin in the central nervous system. The microglia, these are the ones that act like white blood cells. Cool. But I would like you to know the astrocytes, and I picked the astrocytes. I mean, I am a fan of the old school Houston Astrodome, but that's no more. The astrocytes are the most abundant. This is the most abundant neuroglial cell we have in the central nervous system. If you look here, the astrocytes tend to sit between things. So here's an astrocyte, here's an astrocyte, here's an astrocyte. They tend to cling to blood vessels. So here we have an astrocyte with part of its dendrites, part of its tendrils clinging to an axon, and the other part of its dendrites clinging to a blood vessel. So the astrocytes tend to kind of hold things together. And that's one of the functions I'd like you to know, structural framework. So the astrocytes kind of go between things, blood vessels and axons. The astrocytes hold things together. You can see how that would give it structure. And the other function I'm going to point out would be repair damaged tissue. Now we know that there's a lot of, almost all central nervous tissue cannot be regenerated, right? Particularly the neurons. However, when we say here repair damaged tissue, the astrocytes can repair some of the other tissue. The astrocytes can repair some of the blood vessels. The astrocytes can repair some of the other neuroglial cells. And even if the neuron gets damaged, the astrocytes can minimize the damage. So I pointed out those two functions only. You with me? Okay, good. The example of a helper cell I'd like you to know in the peripheral nervous system is the Schwann cell. The Schwann cells are kind of in with this myelin covering. So we're going to discuss the myelin sheath in a minute, but the Schwann cells surround the axons in the peripheral nervous system, and the Schwann cells form the myelin sheath. That's pretty important. So most of the axons in our peripheral nervous system, most of those axons have that fatty covering. And this fatty covering is called the myelin sheath. The myelin sheath is formed by the Schwann cells. Nice and straightforward. Speaking of the myelin sheath is a fatty covering over the axons in the peripheral nervous system. Here's an up close look at a myelin sheath, and you can see the Schwann cell nucleus. Cool. Most axons in our peripheral nervous system are myelinated, almost all. If I go back a slide, you can see that the myelin, it kind of forms these bubbles. It's not continuous. Pretty easy to see. The spaces in between the bubbles of myelin are called the nodes of Ranvier. The spaces in between those bubbles, the nodes of Ranvier. We're going to talk about why those are there a little bit later. Cool. When we were in lab on Monday, so when you're in lab tonight, we opened up the sheet brain and we saw that there was gray matter and white matter of the brain tissue. Well, it gets that coloring or that look because of whether that brain tissue is either myelinated or unmyelinated. So the gray matter, the little darker tinted tissue in the brain, gray matter, is unmyelinated. But when we have the presence of that myelin covering, it gives the appearance of a little bit lighter brain tissue or white matter.
What creates the myelin sheath in the second?
The oligodendrocytes.
Oh, sorry.
That's okay. So if you look down here, here's a real image of a nerve, a peripheral nerve. Look at this one. Here's the diameter of the nerve. Here's the myelin covering. Look at how thick. Wouldn't that give it a lot of protection? It sure would. Versus on this side, if we just look at the peripheral here, this is the outer covering of the nerve, thin, unmyelinated. So we can see the benefits of that myelin covering. Now, can't we? Yes. Okay. Getting towards the end here of the anatomy. So let's go back for a moment to the neuron. We remember that there's two different components of nervous tissue. We have the nerve cells or neurons, and we have the neuroglia or helper cells. That sound familiar? We have the neurons or nerve cells, and we have the neuroglia, the helper cells. Cells. If we go back to the nerve cell, there's actually three different types of nerve cells. The nerve cell that we identify with the cell body, the dendrites, and the axon, that is what a typical motor neuron looks like. So the structure we just learned is actually a motor neuron, which means nerve impulses are traveling in what direction? Away from the brain or towards the brain? Away. I just want to point out that sensory nerves and the neurons inside the central nervous system, the interneurons, look a little different. You don't have to know the specific differences. All I'm pointing out is that there's some structural differences between the sensory neuron and the interneuron in the central nervous system. What we talked about was the motor neuron. That's it. Has it been a long week for everybody? I know. So all I'm going to ask you to know is this. I'd just like you to know that the sensory neuron, I don't care about the structure, but the sensory neuron is sending information from the periphery to the brain. The interneuron is processing and sending information inside the brain. And then the motor nerve is sending information out to the body. I can do the same thing here. The sensory nerve, looks a little different, but that's fine. The sensory nerve functionally is sending information towards the brain. And that sensory nerve will synapse with an interneuron in the spinal cord and brain. And then the interneuron will eventually synapse with a motor neuron. And the motor neuron is sending information out to the body. That's all. No, the Schwann cells are only in the peripheral. All right. So we're about to talk about the nerve impulse. I could not be more excited. It's true. But I feel like you need a little pick me up, and I've got just the thing. I was gonna try and put it on the big screen, but because I can't figure it out. So I just want to show you something, little levity. I went out for a bike ride last Sunday because it was like 45 degrees, which is within my wheelhouse. And I'm riding out near Averill Park. Okay, and this is what I see on the side of the road. A free toilet.
Saw that the other day.
Did you?
Not that one, another
You
one.
saw another one?
Yeah, there are two of them. I was putting one. Free.
So as I'm going by, I thought it wasn't even clean. So my friends were like, to use or to take. You know, sometimes when you're out on a long bike ride or a long run. It'll be kind of fun. Like right on the side of the road.
So it's just, I went to college this last year with my bachelor's, and there's literally a guy that has a toilet garden.
The toilet
So,
garden?
yeah, he like collects toilets and not like plants in them, like literally there's probably like a good 50 toilets all over his lawn. Yeah. They got so bad that the county like tried to sue him and everything, but he won because it's his property and he owns the house. So he doesn't owe anything on the mortgage. But yeah, it's like just toilet. Yeah.
Yeah, I can't get you excited about neurons. We're talking about toilets. This is what happens. All right, so we're gonna spend the rest of tonight talking about these nerve impulses. Okay, I get excited. So I just want to remind you, you see this here, highly irritable. Excitability was one of those characteristics of muscle tissue, remember? Muscle tissue is excitable. Well, so is nervous tissue. Nervous tissue is even more excitable. We tend to use the word irritable, which is kind of funny. So it's excitable. Nervous tissue and muscle tissue are responsive to stimuli. So before we start a discussion on how a nerve impulse is generated, we're going to have to review that resting membrane potential. What kind of feelings does that bring back to you, everyone? Does that feel good or? So we're going to review all of this because this is the reason why we're able to make an electrical current, because we have this resting membrane potential. Do you remember? What this means is at rest, a cell is negative on the inside compared to the outside. Does that sound familiar?
Yes.
At rest, every cell in the body at rest, the inside of that membrane is negative relative to the outside of it. We're going to start to add some numbers. For a nerve cell, for a neuron, that inner charge is -70. And this would be true along the cell body. This is also true along the axon. Different types of cells in the body have different negative charges. Muscle cells tend to be more around -90 millivolts on the inside, but a nerve cell is -70. And what did we say about this resting membrane potential? It is a form of potential energy. Right? Even though at rest, the cell isn't doing anything, the fact that it has this negative charge is a form of potential energy. I gave the example way back when of a battery. You know, a battery, when you hold it, it doesn't do anything, yet it contains that potential energy. So it is this potential energy that gives us something to work with. You see, it wouldn't be possible to generate an electrical current from nothing. So how do we generate this charge? Ions, right? What ions? Potassium and sodium. So it's important that we review this because we're going to continue to talk about these ions. So we know that along the cell membrane, we have these protein channels, right? Which ion is found in higher amounts inside of the cell? So we have more intracellular potassium, and we have more extracellular sodium. Hopefully this at least sounds familiar. Yes? I did my job, and you did your job. So, aren't these ions going to diffuse along their concentration gradient? Of course. Sodium will diffuse from the outside of the cell to the inside of the cell. Potassium will diffuse from the inside of the cell to the outside of the cell. And we remember that each ion has its own channel, correct? Millivolts. Millivolts. I'll come back to that. So how is it that we can get a negative charge on the inside when both ions are positive? Potassium. Correct. So remember, potassium diffuses out faster. So I just gave you an example, right? We just applied some numbers. They're made up numbers, but it allows us to see the principle. I said for every 1 millisecond, maybe the inside of the cell loses 10 potassium ions. So from the perspective of the inside of the cell loses 10 potassium ions. In that same unit of time, maybe we only bring 3 positive sodium ions in. So from the perspective of the inside of the cell, it has lost 10 positive ions. It has only gained back 3 positive ions. What is the resulting charge? So it's simply because potassium diffuses out faster and sodium can replace it. Oh, I'm excited. This is one of my favorite lectures all semester. I don't care if it's dark at 4:30 in the afternoon. I don't care if we're all tired. And then we took it even further. We said, if we let this continue, do you remember? If we just let this passively continue, wouldn't the body find a way to find homeostasis? It absolutely would. The body's so good at it. If we just let it be like that, the body would find some way to change this. because your body doesn't want a difference. But in this case, we want to keep the difference, don't we? So we have to have a way to keep this possible. So we have the sodium potassium pump. Everyone's favorite. So at another channel, we constantly pump potassium in. For every 1 ATP, we pump 2 potassium in and 3 sodium out. And the goal of the sodium potassium pump is to maintain the original concentration gradient. The goal of the sodium potassium pump is to maintain the original concentration gradient. So even though potassium is passively diffusing out, even though sodium is passively diffusing in at different speeds, whatever, we're also constantly pumping potassium back in, sodium back out. We always want to have more potassium inside. So this pump constantly pumps potassium in to make sure that there's always more potassium on the inside. We always want more sodium on the outside. What does this pump do? Constantly pump sodium out to make sure we always have enough potassium on the outside. Because if we have those original concentration gradients, if we always have more potassium on the inside, if we always have more sodium on the outside, then potassium will diffuse out faster than sodium, and we'll give that difference in charge. No, it just depends. You gonna make me sing a Carpenter song? We've only just begun. Karen Carpenter? She's amazing. Someone knows Karen Carpenter, right? Okay. I have this is gonna sound lame, but I have the Carpenter's Christmas album. Love it. It's a very good one. Even though I'm not playing Christmas music yet. You know, some people are. I don't think you're alone. Okay, so this forms the foundation. Understanding that at rest, the inside of the cell is negative compared to the outside. Understanding that this gives us potential energy. And lastly, remembering that this is all possible because of the movement of ions. Because once you understand this, to go from this to a nerve impulse is actually quite simple. I'm going to blow your mind in a minute. But first, we have one order of business. We've been mentioning these protein channels. Well, it's important now that we address something about these protein channels. They can be opened or closed based off of different things. So there's 3 ways that a protein channel can be opened or closed. And we keep this really simple. A chemically gated protein channel. It's way past that. Yeah, there it is. A chemically gated channel means that that protein channel opens or closes based on the presence of a chemical. If a certain chemical is there, the gate will open. If that chemical is not there, the gate won't open. An example of this is when acetylcholine crosses the synapse into the muscle. We didn't talk about this specifically, but we will later in this chapter. When acetylcholine crosses the synapse into the muscle, acetylcholine binds to this channel. And it is a match made in heaven, because when acetylcholine binds to this certain channel, it opens. That's an example of a chemically gated channel. That protein channel opens or closes based on the presence of a certain chemical. An example is acetylcholine at the neuromuscular junction. I don't think it's so bad. Then we have voltage gated channels, which simply means that a protein channel will open or close based off of the voltage. So as ions flow in and out, depending on where this is, if the charge reaches a certain voltage, boom, channel will open. Usually it's based off the charge. I mean, they're relative to each other,
Okay.
right? So this is talking about, this picture is talking about the inside, even though the number here is outside, because at rest, a nerve cell is -70 on the inside. So they're trying to show the arrow. An example of a voltage gated channel is in the neuron for a nerve impulse. So we're going to talk about this today. When we go from this to a nerve impulse, those channels will open or close based off of voltage. It reaches a certain threshold, a certain voltage, gates will open. That's a voltage gated channel inside. And lastly, there are instances where we have a mechanically gated channel, where a protein channel will open or close based off of mechanical force, such as in the skin. So if you apply certain pressure, channel opens. Pressure removed, channel closes. I wish all images were this clear. Okay. Yeah, so this just means that a gate will open upon the presence of a certain amount of pressure or force. An example would be the skin. But we're going to focus on the voltage gated channels. And that's what's happening in the nerve cell during a nerve impulse. Good. Oh man. Oh dear.
I'm excited.
I'm going to work it up here. Sorry. All right. So let's introduce the nerve impulse. It turns out that there are two different types of impulses. There is what we know of as a nerve impulse. That's the action potential. And those are some of the terms I was using in the muscle chapter, remember? So an action potential is a full-blown nerve impulse. Bless you. The action potential is a full-blown nerve impulse. And when I say it's a full-blown nerve impulse, that means that the nerve impulse travels the whole length of the axon. It doesn't matter if that axon is this long or this long. We have axons that the cell bodies in your spinal cord, and the axon goes all the way down to your pinky toe. So an action potential means that we've got an electrical impulse traveling the full length of the axon, no matter how long the axon is. So when we think of nerve impulse, you're usually thinking of an action potential. But you see, there's also what's known as a graded or local potential. And that is like a mini impulse. So a graded or local potential is more of a mini impulse. It only travels short distances. That's why it's called local. It doesn't have widespread effects. A mini impulse, a local or graded potential, travels a short distance.
Send out the signal from.
We're gonna get there. Bear with me. So what I want to do first, before we put it all together, is I first just want to introduce some of the major differences between these two. Because, and I'll just jump to this because Nicole is so eager, it gets me excited. What we're going to find out is, in order to make an action potential, which travels the whole length of the axon, we have to create a graded potential first. So what we're going to see is your neuron will make a mini impulse that goes from here to here. And if that graded potential, if that mini impulse reaches a certain threshold, then the action potential will fire. So we send a lot of graded potentials. We send a lot of these mini impulses. Not every mini impulse leads to an action potential. Only the mini impulses that are at a certain threshold. But no matter what, in order to make an action potential, which is a current that goes the whole length of the axon, we have to make a graded potential first. Does that answer your question, kind of? Yeah. I'm going to deliver 3 words that change your life today. 3 words. All right, so before we talk about the specifics, I want to just introduce the basic differences between a graded potential and an action potential. We'll start at the bottom. Many cells can make this little mini impulse, not just muscle and nerve cells. So many different types of cells can generate these mini impulses. Probably one of the best examples of this mini impulse would be glandular tissue. So cells within a gland tend to communicate with each other by sending these mini impulses. So many cells can generate a mini or graded or local potential. But for that full-blown action potential, I mean, we're really only talking about a nerve cell. A muscle cell can continue the action potential from the neuron. So we tend to categorize muscle cells as being excitable in this category also. But that nerve impulse, the action potential came from the nerve. So this full-blown nerve impulse that goes the whole length of the axon can only really be generated by nerve cells. Excellent. Let's skip up to this one. In a graded potential, the effects are local. That just means that the impulse travels a short distance. Versus an action potential, which can travel a long distance. That graded potential only travels a short distance. Whereas the action potential can travel the whole length of the axon. All right, check it out. What we're going to find is that the action potentials, I know you're going to hate this, but The action potentials have rules much like the motor unit. You thought you were done with that, didn't you? So the action potential, once it fires, just like a motor unit, each current is the same intensity. I think it's going to make more sense once we go through the whole thing. I just want to introduce this to start. So an action potential, once we get to a point to fire it, that current is always the same intensity, which is different from a graded potential. And that's why it's called graded, because the initial impulse is strong, and then the further the impulse travels, the weaker it gets. And that's why it's called a graded potential. The intensity starts off higher and it goes down. All right, good. So here's what I want to do. I know we don't have much time left because I want to give you your exams back, but I want to introduce the basic steps of a graded potential. Okay. All right. Excellent. All right. Are you ready for those 3 words to change your life? Winning lottery. All we have to do to go from resting to making a nerve impulse, no matter if it's graded or an action potential. Open sodium channels. This is it. That's it. Open sodium channels changes everything. I'm going to show you. So at rest, right? We always start from resting membrane potential, where the inside is negative compared to the outside, right? The inside is negative 70 millivolts. Our first step towards generating a current, no matter if it's a graded or an action potential, is something called depolarization. When we start, the membrane is polarized. It's different. You know, polar ends, north and south pole. The politics are very polarized. Either you're on this side, so it's different. So when we depolarize the membrane, we begin to make it less different, which means that this is going to go from negative 70 to negative 60 to negative 50, negative 40, negative 30, negative 20, negative 10, 0, even going to go positive. So the first step is something called depolarization. How we do that? Open sodium channels. Don't we have more sodium on the outside? Yes, we do. Don't we have more potassium on the inside? Yes. So all we do to depolarize the membrane is we open extra sodium channels. Instead of having only 1 sodium channel open, we open 20. I'm not sure that's an exact number, but I'm just saying it's a lot. Open more sodium channels.
Does the potassium sodium pump also stop?
No,
Okay.
that keeps going over here.
Okay.
But that's fine because this is powerful enough in that instant to overpower it. So by opening sodium channels, and at the same time, closing potassium channels. So we hold potassium in, and we have a sudden influx of sodium. Sodium rushes in. So we have a sudden influx of sodium ions that eventually turns the inside positive. So we bring in that instant, so much sodium, the inside goes positive. So we went from -70 on the inside to +10. How many millivolts changes that from -70 to +10? 80, -70, -60, -50, -40. So in an instant, we have a 80. We have an 80 millivolt spark. In the real world, like in electrical, 80 millivolts isn't much. I'm sure electricians probably get that when they're at work every day, 80 millivolts. But in our cell, a teeny tiny cell, that is significant. Okay, so we open sodium channels, positive ions rush in, turn the inside positive. The next step is the spread of that depolarization. So we open sodium channels here. Well, now we're going to open more sodium channels here. I'm gonna use the analogy of a wave. You know what a sporting event? So when one person depolarizes, imagine the wave. At resting, they're sitting down. During depolarization, they stand up. When you do the wave, when you stand up, doesn't it trigger the next person to stand up? Which triggers the next person to stand up? Which triggers the next person to stand up? So that spread happens where after these channels open, these channels open. In the case of a graded potential, it spreads both ways. So it spreads to one side, but it also spreads, to another. So imagine a wave going both ways. So we depolarize by opening sodium channels, which then causes adjacent sodium channels to open. And that spark, that quick 80 millivolt change is transferred to this. But then, right after an area depolarizes, and the area beside it depolarizes, that area repolarizes. To repolarize is to go back to where you were. If we open sodium channels to depolarize, how do you think we if we open sodium channels to depolarize, how do you think we repolarize? Close them. Because of changes in voltage, because they're voltage gated. So as these ions start moving around, the membrane starts to change voltage. And that change in voltage, even a little bit, triggers these to open. So that's where I give the analogy of the wave, where at rest you're sitting down, open sodium channels to depolarize and make a spark, triggering the next areas to stand up and depolarize. But then after they depolarize, I sit back down. Because there's a stimulus that gets some of these sodium channels moving more. So it has to be an initial stimulus, which just means that we start to bring more sodium in to begin with, which will change the voltage a little bit. And when that voltage changes to just the right amount, floodgates open. Correct. Correct. So this is showing you along an axon. At rest, inside is negative, outside is positive. We have a stimulus, sodium gates open, sodium rushes in, turns the inside positive. Depolarization. Then the adjacent membranes depolarize. And in a graded potential, it spreads in both directions. But after it depolarizes, it'll return back to negative. Just like a wave. Each time, imagine a wave. All right. I forgot to grab something from my office that I want to give you. So I'm going to go grab those and then I'm going to give you your exams back and hand out the final exam review sheet for you. So you'll have it. I have one.
Thank you so much. You're welcome. I don't have practice.
I'm not trying to overwhelm you, but if you want to start pecking away, this is a nice roadmap because it gives you the most important concepts from each chapter. I give it to you now just in case you want to start pecking away over the break. But it's okay if you don't want to or can't also.